Today's lecture focuses on cytokines. Up to this point throughout the semester, we have referred to cytokines as signaling molecules that play a role in the immune response. We've sort of lumped them together collectively as a group of molecules, and we're going to focus on them in a little more detail today. I may have mentioned previously that there are over 200 distinct and different cytokines that have been identified in humans. And if we focused on every single one of those, we could fill up an entire semester worth of material talking about individual cytokines. So what we will do is we will talk about them again as a group and then talk about some distinct categories later on in the lecture. The first point I want to bring up is the term cytokine itself. The root cyto comes from the Greek meaning cell, and kines comes from the Greek kainen, which means to move. So these are molecules that were thought to play a role in making cells move. We've discussed specifically a subset of cytokines known as chemokines, and we will talk about them a little bit more today. But chemokines are a category or a subset of cytokines that do in fact do that. They act in a way to cause a cell to move. Cytokines, for the most part, are protein molecules, but some that have been identified as glycoproteins, which just means that there's a carbohydrate linkage to a protein component. They are low molecular weight, meaning that they are relatively small molecules altogether. Their job is to regulate the intensity or duration of an immune response. How specifically do cytokines do that? Well, there are a number of ways. They can stimulate changes in gene expression patterns. They can regulate proliferation, differentiation, and maturation of certain leukocyte types. They can regulate cell activity, such as allowing for phagocytosis to occur, allowing for the extravasation mechanism to take place, and so forth. They can also play a role in regulating apoptosis. So you can see that they do in fact have many functions in the immune response. They do have an effect on a variety of different cell types. The number one cell type or category that they affect are going to be lymphocytes, but we know that cytokines play a role in having effects on other immune cells as well. And if you do look at the Appendix 2 in the back of the book, this does list those cytokines that have been identified in humans if you're interested in looking at some specifics of cytokines. Cytokines can be described or categorized in many different ways. One way to categorize a cytokine is the type of action it actually produces, meaning how does the cytokine behave? There are 3 different categories that describe types of action or modes of action for cytokines. Autocrine is the first one, which is a condition which the cell acted on by a cytokine is the source of the cytokine. So in this particular example, if you look at the figure associated with an autocrine function, you have a cell produces the cytokine, and the cytokine acts on the exact same cell that produced the cytokine in the first place. The second category is paracrine function, and this is where regulatory secretions arrive by diffusion from nearby cellular source. So in this particular case, you have a cell that is producing the cytokine. That cytokine travels a short distance, typically by diffusion, to then have an effect on a nearby cell type. Typically, this is in relative close proximity of each other or one another. Most cytokines act in either one of these 2 ways, meaning that they are either autocrine or paracrine, and they typically do work at very short distances. There are, however, some cytokines that are considered endocrine, and these are going to be those that pass from the producer cell, again, the cell that is actually producing the cytokine, to have an effect on some target cell that is a further distance away. And because it is a further distance away, the cytokine has to actually enter into the bloodstream, into the circulation of the host, to get to that target distant cell. As you can imagine, again, this does take a time lag associated with it because the cell that produces it and the distance traveled needs to be accounted for. And again, most cytokines do not act this way, but there are some that do happen to fall into this particular category. A variety of cell types are known to be able to produce cytokines. The principal producers, however, are leukocytes. We did discuss very briefly in the first unit of material, in specific the innate immune response. When there is a breach in the physical barrier, those cells that have been damaged in that breach, those epithelial cells, do in fact have the ability to produce a subset of cytokines. So again, it's not just leukocytes that produce cytokines. However, for the most part, leukocytes are the ones that are producing cytokines. And then within leukocytes, there are principal cytokine producers. Obviously, the helper T cells, like we've discussed, that's their main job, is to produce cytokines. However, dendritic cells and macrophages also produce a significant amount of cytokines. Now, that doesn't mean that other leukocytes are not producing cytokines. They do, but these again are the subset of principal cytokine producers. If you look at this particular slide, you will see what again looks like a really rather complicated mess overall. And my point here again, when I do this, is just to show you that there is a lot of complexity involved with this particular topic as well. With this slide, what this diagram is trying to show is specific cell types. In this case, these are all leukocytes that are on here. And what you see listed are cytokines that they produce, and then the cell type that those cytokines are acting on. So again, you can see it's a very complex network of interactions that occur by cytokine production and the cells then that the cytokines are acting on. We can, however, also discuss broad categories of biological function associated with cytokines. The number one component that we have discussed, both with the innate immune response and a little bit with the adaptive immune response, is inflammation. So specifically, with more detail, we looked at the innate immune response and how inflammation is so important in that process. Histamine release, vasodilation, vascular permeability, changes in cell adhesion molecule expression and function, chemotaxis, phagocytosis, all of these things are all part of the inflammatory response and is a key or principal biological function of cytokines. We do know that cytokines play roles in other aspects of the immune response. For example, we know that cytokines play a role in the adaptive immune response, specifically playing a role in targeting the death of altered cell cells or the destruction of altered cell cells, both with cytotoxic T cells as well as natural killer cells acting in that particular capacity. However, we know that cytokines also play a role in hematopoiesis, that is the production of distinct and different types of blood cells, in particular, in this case, leukocytes that are needed for a specific response. And lastly, a key biological function of cytokines is to play a role in promoting activation, differentiation, and proliferation of specific cell types, including T cells, B cells, macrophages, dendritic cells, natural killer cells, and just about every other type of leukocyte. So cytokines definitely play a really big role in allowing for that activation, differentiation, proliferation of these cells. With the next series of slides, I'm going to focus on what I refer to as cytokine attributes. These are mechanisms or ways that cytokines can actually behave. So the first one is something known as pliotropy. Pliotropy is a term in this specific case that is referring to a cytokine that has different biological effects on different target cells. In other words, you have one cell that produces a specific cytokine. In this example, we have a helper T cell producing a cytokine known as interleukin 4. That cytokine can go and act on different and distinct target cells. So you have one cytokine acting on multiple cells, which is a pleiotropic effect. In this case, the different cells that are listed here are B cells, thymocytes, and mast cells, and the effects are similar. For B cells, it does play a role in activation, proliferation, and differentiation. For thymocytes and mast cells, it's playing a role in allowing for its proliferation. So the concept of pliotropy, again, just means a single cell type produces a single type of cytokine, and that cytokine can exert its effects on multiple different types of target cells. So the next two attributes are listed here. The first one, the next cytokine attribute I want to discuss is known as antagonism. With antagonism, you have the effects of one cytokine inhibiting or offsetting the effects of another. This is actually part of a regulatory mechanism, a really fast way to turn off function associated with a given cytokine. So let's look at the figure in this example. You have your helper T cell producing interleukin 4. Interleukin 4's job, like we talked about in the previous slide, is to play a role in B cell proliferation, but also can play a role in the switch to produce the IgE class of antibody. However, if that switch needs to be turned off almost immediately, there is another cytokine that can be produced, interferon gamma, and this particular cytokine blocks the activity of interleukin 4. So again, this is a regulatory process to help rather quickly stop the activity of one cytokine with the production of a different and distinct cytokine. The last cytokine attribute that I want to discuss in general is known as cascade induction. And in this particular case, you have the action of one cytokine on a target cell, inducing that particular cell to produce one or more additional cytokines, which then can play a role inducing other target cells to produce different and distinct cytokines. So this is kind of like a mechanism that acts as a cascade to trigger the production of lots of different cytokines. So if we look at this example, up at the very top, we have the helper T cell producing interferon gamma, which in this case is acting on a macrophage. That then triggers the macrophage to produce interleukin 12. Interleukin 12 acts on helper T cells to then stimulate the production of additional distinct cytokines. And so this is the cascade induction, in that one induces another, and eventually you get a whole slew or series of cytokines that are being produced in this step-by-step mechanism. Now, cytokines can also be classified in a different way. We first talked about their attributes, and we talked about their mechanisms or mode of action. However, another way that cytokines can be classified is based on their receptors. Interestingly, you might think that cytokines get lumped together because of their function or the function that they exert. However, that's not really the way cytokines are typically classified. They are classified based on the receptor that they bind to. So this goes back to our conversation or our discussion of receptor-ligand interaction. And we have, in a basic example of receptor-ligand interaction, you have a molecular receptor that is typically, in most cases, part of a cellular membrane that acts to receive a molecule, a molecular signal known as the ligand. And when we did discuss receptor-ligand interactions, we also looked at one specific example of cytokine induction. And so if it might be beneficial if you go back to that particular lecture and look at that signal transduction pathway that occurs with the binding of the cytokine to the receptor and inducing that particular cell to behave in a specific way. So back to the topic here, though, cytokines are classified based on the receptor structure, and there are a number of categories of cytokines like you see here. Interleukin 1 family, Class 1 cytokines, Class 2 cytokines, tumor necrosis factor family, interleukin 17, and the chemokine family. And we'll talk about them here in a little more detail with the next slides. So the first category or classification of cytokines is interleukin 1 family. So recall that it's not the cytokine itself that distinguishes the classification, it's the receptor that the cytokine binds to. So in this particular figure, what you see is the structural representation of the receptor, and in this case you have the interleukin 1 cytokine that would be binding to that receptor, acting as the ligand. In this particular case, most members of this family are in fact pro-inflammatory cytokines. That is, they play a role in inducing the inflammation. There are 11 distinct family members that have been identified. They are usually secreted very early on in the immune response and are produced mostly by macrophages and dendritic cells. This particular cytokine family is stimulated by viral, parasitic, and bacterial antigens. These cytokines can act locally, typically allowing for capillary permeability. They can also act systemically by traveling through the bloodstream and signaling to the liver to produce specific immune response proteins. This family does play a role typically in allowing for adhesion factor expression to occur, which is important for extravasation, and does play a role in regulating the hypothalamus to regulate body temperature. Taking one step back, the name of this family again is interleukin 1 family, and the term interleukin, inter means a means of communication between, and leukin refers to leukocytes. So this particular family is very correctly named a way to communicate between leukocytes. The next category of cytokines are known as class 1 cytokines, also the hematopoietin family. Hematopoietin is a specific cytokine that plays a role in the production of red blood cells and falls into this particular family. Remember again, when we talk about this classification, it is the structure of the receptor that groups the cytokines together. In this case, what you can see on the slide is the structure of the receptor, as well as a number of the ligands that are known to bind to this particular category or class of receptor. This is the largest and most divergent family of cytokines, with approximately 50 distinct members falling into this particular family. In general, these cytokines do play a role in signaling differentiation and activation of specific cell types. They do play a role in allowing for the production of antibodies in B cells, and they are also known to play a role in activating granulocytes. The next group is class 2 cytokine, which includes a specific cytokine known as interferon. There are a number of interferons that fit into this particular family. The term itself, interferon, comes from the fact that these cytokines typically interfere with viral replication and other viral functions. And so, like the slide says here, this particular family does play a really important role in antiviral responses. This cytokine family does play a role in allowing for the increased expression of MHC, and hence also plays this really important role in fighting off viral infections. The next category is an unusual family, referred to as TNF family or tumor necrosis family. The cytokines that fall into this particular category are known to have soluble or membrane-associated components and are very distinct overall. As you can see from the description here, these cytokines are involved in the development, effector function, and homeostasis of distinct cell types, including skeletal, neuronal, and immune systems. So some of the family members here are not only playing a role in immune responses, but can be playing a role in other areas of cellular function as well. The interleukin 17 family is the next category or family of cytokines. And interestingly, this is a more recent addition to our nomenclature or our categorization. These cytokines are typically produced by helper T cells. And what you can see here is that the receptors are made up of lots of different components that can either be homodimers or heterodimers to produce a variety of different receptors in this particular category. Most of the cytokines that fit into this particular category are in fact pro-inflammatory and can be expressed by a number of different cell types. And the last category or classification of cytokines is a family known as chemokines. In this particular case, this is the only family or classification where all of the cytokines that fit into this particular family do in fact have a similar function. All of these molecules are chemokines, which are chemoattractants. They play a role in allowing for cell migration to take place during the immune response. However, again, they're categorized by their receptor structure, and you can see the receptor in this particular diagram is quite distinct from any of the other receptors that we've looked at with the previous classifications. So again, all of the members of this particular group are in fact chemokines that act as chemoattractants to play a role in allowing for cell migration to take place. So at the beginning of the lecture, we talked about different attributes associated with cytokines, and one of the attributes that we discussed was antagonistic function. The antagonistic function is really a built-in mechanism to help regulate cytokine activity and cytokine function. So in this particular slide, the point is that there are many different cytokines that act as an antagonist, that is, inhibit the activity of another cytokine. However, what I wanted to do with this particular topic here is introduce one more concept that we will be looking at in a little more detail in our next unit of material. And that is that there are some situations where pathogens have evolved molecules or proteins that play a role in binding cytokines and inhibiting their function. So what you see in this table is an example of a series of viruses and virally encoded proteins that they produce that can in fact play a role in negating or downplaying the immune response of the host. This then makes the virus a very good pathogen because it has a way of tricking the host immune system into being dampened down or turned down so that the immune response is not heightened. So again, this is what we refer to as an evasion mechanism, and this is what makes some pathogens very successful, is that they have a way to outsmart or get around or get away from the immune system. In this particular case, these are viral proteins that have the ability to act as a cytokine or bind or block the activity of a specific cytokine, again dampening that immune response in the host system.